This is the Anthrobiology Podcast, hosted by me, Gabby LaPera. Every 2 weeks, I interview a biological anthropologist about what makes us, us. Hello, everyone. Welcome to another episode of the Anthrobiology Podcast. Joining me today, I have Dr. Christopher Schmitt at Boston University. Good morning. Hello. How are you?
Good morning. I'm very well, thanks. How are you doing?
I'm really good. Thank you so much for agreeing to come on the show. I'm really excited to talk to you. It's been a while since we've had a primatologist on.
Oh, cool. Well, I'm glad to start a new run, maybe.
Yeah. Yes. I'm trying to think where I want to start. I guess like the first place I want to start is what drew you to primatology in the first place? Like why this path for you?
Yeah, that's actually a really good question because I've got a kind of interesting story about it. When I was a kid, I really liked animals. I was like very socially awkward. So like I used to like to lay on the ground with my dog and like look deep into his eyes and try to understand him, which
Yeah.
is a lot easier than other kids. And I loved going to the zoo. And so in high school, I went to an inner city public school in Milwaukee, Rufus King. And our high school didn't have all that much money, but we did have a wealthy alum who was in the biotech industry, and he donated scholarships through the Earthwatch Institute to our high school, and it was an organization run out of Boston. I think it still is, and at the time they would give scholarships to high school kids to actually be field assistants on field projects,
That's so
which was
cool.
amazing. And yeah, it was super, super cool. So I was one of the six students who won a scholarship to go to the field in the summer between my junior and senior year of high school. And originally, I wanted to go to South Africa. There was a project basically doing censuses of big game, and at the time, I was really interested in zebra behavior and elephants, and it was too expensive. Like I would have had to have supplemented it with money. The scholarship wouldn't cover it. But what wasn't too expensive was a scholarship to go to Argentina and study black and gold howler monkeys under a project that was run by Govindasamy Agoramurty, who has studied a lot of howler monkeys. At the moment, I think he's at the university in Taiwan, in Taipei. And so I got the scholarship. It was my first time out of the country. My dad was a big camper, so I'd been camping a lot, and it just kind of merged all of these things that I loved. Like I was in this like my first time in like a real international group of people, and it was a lot of fun. And pooping outside was fun, and I saw amazing behavior, which nowadays like howler monkeys, I'm kind of like, no offense to people who study howlers, but I'm kind of like, okay, they're kind of boring, right? They just kind of sit there all day. But I saw a mating take place like a foot over my head. We were collecting urine. It was like a really, really amazing experience. And so I was there for a month. I really, really loved it. And when it came time to go to college, I went to the University of Wisconsin because it was, in-state tuition was cheap and I could afford to pay for it. But also Karen Stryer was there. And I ended up taking her primate behavioral ecology class. And whereas I'd been majoring in English literature, and I did major in English literature, I picked up a double major in zoology, and with Karen's help, essentially, like made my way into the field, well, with her letter of recommendation. After college, I still wasn't sure about primatology. And I applied to work as a field assistant on Susan Perry's long-term capuchin project in Costa Rica. Got on that project, won a scholarship to pay for me to be able to get there. And while I was there, I just decided I was like, yes, I'm going to do this. This is pretty great.
I think honestly, like a hint for like you and other people out there is if you are waxing poetic about collecting urine samples and watching monkeys mate a foot above your head, this is probably
Yes,
the field for you.
it probably is. Exactly.
That is so funny. So right up my alley. I don't know if
Oh, cool.
listeners know this, but I really love like energetics and like history theory and all the stuff that
Yes.
you are really, really into. I'm going to read a little bit from your website because you did a really good job writing it. And I think that people should be
Oh,
praised
thank
for good
you.
writing.
It's that English major. The English lit major really pays off.
Yeah. It totally does. I really appreciate it. I'm an editor slash writer in real life. So it's like literally all I do all day is read other people's writing. development and life history in non-human primates as they relate to adaptations to extreme ecologies, which are becoming more prevalent and severe with anthropogenic climate change. That is like what you specialize in. There's a lot of words in there that I think are worth breaking down. First, I think, can you give like a quick overview of life history theory? This thing that people spend so much time talking about?
Yeah, okay, quick overview. So, life history theory, as I understand it, is the study of the ways in which organisms kind of evolve to have various set points that represent energetic trade-offs between different phases of life. So, for example, like how long you gestate, like how much of you is developed when you're born, how old you are when you are weaned, how long it takes for you to reach sexual maturity, how many offspring you have within a certain time period, and when you senesce, and whether or not you go through menopause. I think all of this is kind of the domain of life history theory, and really kind of where life history theory happens is energy and how much energy you're able to sequester, whether or not you need to exchange energy between different bodily processes, and what that does to these different set points for when you transition from one phase to another.
Yeah. And all of your work, like most of your work, because you're also doing some stuff about like conservation and like range and stuff, which feeds into this as well. But it all is kind of taking like the grand theme I see is taking like development and life history theory and using that to understand outcomes for individuals over time.
Yeah.
And you've done this in a variety of ways. I'm trying to decide. I think we should talk about one of your major research populations so people can imagine it in their heads as we're talking about it.
Sure.
You study African green monkeys / vervets. What do they look like? Where do they live? Are they cool?
Yeah. I mean, I think they're pretty cool. They're kind of what I call garbage monkeys, and not because they're terrible. I mean, maybe they're a bit trashy. They are a bit trashy, but I call them that because they can live almost anywhere off of almost anything. They're very scrappy. They're really well adapted to savanna environments. So you find them throughout Africa, south of the Sahara. They don't do quite so well in forests, although my colleague and friend Kate Detweiler discovered a vervet species that is kind of living in the Congo Basin, and it's debatable whether it's basal or whether it's nested. Like the genetics are kind of ambiguous about it, but I think that they're basal to the savanna monkey clade. And like I said, they're very scrappy, so they're very widely distributed. They're very successful in anthropogenically impacted environments. So like they love garbage, they survive well in cities, they are very adaptable.
Like
Erica
pigeons.
van de Wal, like pigeons. Yeah, Erica van de Wal is a colleague who works in South Africa on vervets and has done all these cognitive experiments. They're cognitively very complex. They have cultural traditions. They learn really ably through social learning. They're really kind of scrappy, smart species. And they also have, like I mentioned, a really wide kind of ecological distribution, which kind of reflects that adaptiveness, including in the Caribbean. So there is a population of like a lot of populations of vervets that were brought over during the transatlantic slave trade into the Caribbean and kind of took root on several different islands. So I've worked with vervets on St. Kitts and Nevis. There's vervets also on Barbados. Just in the last decade or two, they have since spread to St. Martin. So they're kind of making their way through the islands, and they do very, very well there. So we've studied them there as well.
Is that humans spreading them still, or is that them just being crafty? Getting
I don't know, to be honest. Like, I don't know if somebody like saw vervets on other islands and were like, oh, those would be a fun pet. Like that is something that happens quite a lot. And maybe they brought one or two to St. Martin and they got out. It's also possible, I think, for them to stow away on a boat. I mean, I'm not saying that's what happened, but I could totally see that happening. I'm not sure. I don't know how it happened.
No, it's definitely really interesting. I was in the Galapagos last year, and they said that like one of their major problems was rats, because before humans started coming to the Galapagos, there were no rats there. There was no way for rats to get there.
Yeah.
And so they have like a lot of ground nesting birds, and the rats love to eat those.
Just decimating them.
I also had never thought about the idea of a ground nesting bird. Some of them nest, I think, is a very strong word for what they do. It's more of just standing next to their egg on the ground. We're not here to talk about birds. So you study these vervets. It looks like you study them at Wake Forest, and then you also study them in the wild.
Yes. So this is part of a larger kind of research structure that my mentor and my postdoc mentor, still my mentor, Trudy Turner, amazing scholar, that she kind of founded with Nelson Freimer, who was also a mentor of mine when I was at UCLA, and David Yench when he was at UCLA. This is a really great research structure wherein we've got like all of this variation across Africa south of the Sahara that we've kind of sampled vervets from. But then we also have this bottleneck that happened 400, 450 years ago in that travel to the Caribbean. And then in the mid 70s, Lynn Fairbanks and some colleagues founded a vervet research colony at UCLA from a population of monkeys from St. Kitts. And so we've got these two bottlenecks, and then the captive colony is very well pedigreed. And what this means is that we have like a lot of statistical power to find connections between genetic variation and other phenotypes in the captive colony. But we also can kind of scale out and then go and look for how that variation plays out in a relatively inbred population like St. Kitts and Nevis, or in very particular kind of ecological circumstances across Africa. So it's a really nice kind of way of making these kind of genotype phenotype connections around a lot of different traits. And part of it is an accident of history, and part of it is just, really good planning on the part of Trudy and colleagues.
Yeah. And for listeners, just in case, a population bottleneck is when you have a population that gets really small for whatever reason for a little while. And when that happens, that changes the percentages that you see in terms of like genotypes.
Yeah.
And so that means that you'll end up with these like really weird random small populations where like some random disease is much more likely to happen, or like, I don't know, the monkeys have red fur for whatever reason, just because like those were the ones that were around. It's super interesting. It happens in humans too.
Mm-hmm.
But can you describe what a vervet looks like?
Oh yeah, sorry. So they are usually kind of like a grayish brown color in their body, like not super remarkable, kind of, a little bland. And then they've got these cute little black faces that are usually surrounded by maybe a white ruff. And then, like, the top of their head is that kind of grayish brown cap. The tip of their tail, they usually have a long tail that's a little bit longer than their body length. And the tip will be black or gold, depending on where in their range are. And they're about the size of like a small dog or a large cat, I would say. They're sexually dimorphic, which means the males are usually about 1.5 times the size of the females, although it depends on where in their distribution you're looking, because the dimorphism varies. And another kind of cool thing about them, and one of the reasons why people maybe know them better than some other monkeys, is they have their scrotum is bright blue and their penis is bright red. And for the females, the vulva is not as bright, but also blue, and the clitoris is kind of pink. So they've got this like interesting coloration that's like exaggerated in the males that we think maybe is a sexually selected signal. Like maybe it has something to do with how females are choosing mates. But like there've been captive experiments where somebody literally spray painted their scrota different colors of blue to see if females liked one more than the other, and females just liked whatever was the most different. So we're not quite sure what's happening there, but we see variation. So in Southern Africa, where I work, it's like a kind of bright turquoise color. And then if you go to East Africa, it's a little more green. And then if you go to West Africa, it's kind of blanched out into kind of white or very, very pale blue. This is something that changes developmentally. So when they're born, it's kind of a periwinkle color for everybody. And then as they reach sexual maturity, the color starts to get more kind of sex specific.
Huh. That's really interesting. I have a lot of thoughts about that, mostly about that study with the pigeons where they found out that the female pigeons began selecting for male pigeons with the tracking bracelets on them.
Oh, I didn't hear about that.
Yeah. Apparently, like the female pigeons were like, I love that jewelry. Like, I'm just so into it. And so, like, they accidentally biased the sample really significantly. But no, so you've studied these vervets. You had this really excellent study that I thought was really interesting called obesity and obesogenic growth are both highly heritable and modified by diet in non-human primate models and the African green monkey. I think I missed an article in there, but really good article.
Thanks.
In science, titles are exactly what the thing's about. No surprises. But you looked at a bunch of vervets. I did have a really fun time imagining you guys taking all of these measurements because you did a lot of body measurements, like crown to rump length and BMI and waist circumference. And I just imagine like someone measuring these little. Were they cool with that?
Well, they were because they were all kind of in a K-hole. So they didn't really mind in that moment. So like
sense.
that study was done at the VRC in captivity, although we've done the same measurements in monkeys in the wild as well. And in the VRC in particular, because of FDA requirements, the monkeys need to be tested for TB 3 times a year, tuberculosis, just to make sure that they don't have it, because obviously it's very contagious. And if it gets into a primate colony, a lot of monkeys will die. And the way you test them for TB is you actually inject a little tuberculin into their eyelid because that skin is very thin. And so they will have a very clear reaction if they are infected with tuberculosis. And of course, it's not easy to inject something into somebody's eyelid when they're awake. So 3 times a year, in order to get tested, they do need to be anesthetized. And usually it's ketamine, sometimes medetomidine. These are just general veterinary anesthesia drugs. And when they're usually under for about 20 minutes. And so we also do basic kind of health checks when that happens. And when I was a postdoc at UCLA, they were like, well, why don't you go for one of these collections and think through what might be an interesting study to work with a captive colony? Because I had, at that point in my postdoc, done, about 2 years of field work with the wild monkeys. And I was trying to think through, what my own personal study was going to be. And while I was at the colony, I saw them taking these measurements. So there are people that take them. I didn't necessarily have to take them through all 20 years of data that we were digging through. But yeah, when I was there, they're anesthetized, they're very easy to work with, very delicate with them when they're coming out of the anesthesia. And the measurements are standardized. So yeah, the colony collects them. I collected some more specific measurements that I was particularly interested in that came out of an NSF workshop called Bones and Behavior, where we're trying to standardize the ways in which we measure all of the primates and humans in order to make everything comparable. And yeah, it is really fun to measure them. But I noticed when I was measuring them, particularly for waist circumference, I was like, there is a lot of variability here on how much they have around the middle. And for context, the vervet monkeys in captivity, and just a vocabulary kind of note as well. I'm calling them vervets and green monkeys interchangeably, but green monkeys refers to the vervets that have their ancestry in West Africa and vervets in Southern Africa. But people have used both as kind of general terms for all of the genus Chlorocetus. But in this case, with the green monkeys, yeah, they get a lot of abdominal fat when they put on a lot of weight. And there were some monkeys that were very, very, very heavy and had these little pooches that would drag on the ground when they were actually awake and running around. Then there were other monkeys that were perfectly lean. And that was where the question for this paper came from. I was kind of like, well, why are some monkeys putting on so much weight and not others? And, the kind of knee-jerk idea is that, well, maybe it's related to dominance rank. But when we dug into the data, that didn't seem to be the case. It didn't seem like the dominant animals were getting that much priority of access to food that the subordinates couldn't eat. And some of the individuals that were really heavy were actually low ranking. And so it gave me the idea that maybe this is something that has to do with genetics. Maybe there's predisposition, like what we see in humans or what we think we see in humans. But then also I was wondering about it developmentally because none of the young animals were very heavy. And in fact, with the younger animals, I struggled to see much of a difference. So clearly this was something that happened sometime during development. So I was really interested in understanding that interplay between, body fatness when they actually become obese. Is this a growth trajectory that happens to some individuals and not others? And can we kind of break it down between all of these different factors? I also was really lucky in that the VRC was a location where they'd done a lot of dietary experimentation. So I didn't myself have to give the monkeys a challenge diet or anything like that. It turned out that in 2004, so for context, I was doing this work in 2010 through 2015. In 2004, the monkeys were kind of getting a little heavy universally. And so for their health, they switched their diet over into a high fiber, high protein diet to try to get the whole colony to lose weight.
Did they like
And
that?
they did not. They hated the diet. Like they hated how it tasted. Like they didn't like it. So they just didn't eat. So it was actually a period during which the whole colony, on average, lost about 10% of their body weight,
Wow.
which is substantial, right? So, like, for context, I weigh like 180 pounds. That'd be like me losing 20 pounds in a couple of months. It's like really dramatic weight loss. And that, kind of plugged me into a whole literature of trying to understand what happens when humans face huge dietary challenges. So, for example, with my students, I always talk about the Dutch hunger winter. So, during World War II, when the Netherlands was cut off from food by Nazi forces, like various parts of the Netherlands had almost nothing to eat for several months at a time. And, 50 years later, when you look at metabolic disorder in the Netherlands, you see a high prevalence of it in those areas where food was cut off during that time period, suggesting in part that the individuals who were gestating inside of their mothers during that time period actually were evoking kind of a lifelong metabolic cost to that food deficit while they were in utero.
Yeah.
Yeah.
We're like kind of dancing around the idea of epigenetics. So
Yes. Yes.
let's just talk about epigenetics.
Let's just talk about epigenetics. So yeah, in this study, we were using mostly quantitative genetics methods. So essentially trying to suss out how much heritability in these traits or how much of the genetic component of these traits is controlled for by, how much is genetic and how much is environmental.
Mm-hmm.
And that environmental component also includes what we call maternal effects, where we can look at multiple individuals who are born from the same mother and see if they have a commonality in how these traits are expressed later in life. And what I found in that study in particular that was really interesting was, first of all, that there did appear to be a genetic component to whether or not animals became obese when they're older. And not just that, but also whether or not they showed a developmental trajectory in the growth of their body weight or their BMI that led to being obese when they were older. On top of that, there was like a very significant component, not just of diet, but of what their mother was eating when they were gestating to whether or not they became obese when they were older.
So that time when they switched the diet over, that was like the Dutch hunger winter for the Vervets.
Essentially, yes. Yeah.
And so you were able to compare like the offspring from that time versus like the before times when they were eating the diet that they liked.
Yeah, exactly. And they actually subsequently changed back to the regular monkey chow diet because the monkeys disliked that diet so much in part, but also because the colony moved from UCLA to Wake Forest, and when they were in Wake Forest, they changed them back to this chow diet. So yeah, there was a really nice way of comparing, like, was this monkey gestated on one diet? What outcome do you have in their growth trajectory and also their, adult obesity phenotype? And it did appear that those monkeys gestated on that challenge diet in particular were more prone to becoming obese as they aged. And this is again like what we see in the Dutch hunger winter, where we've got increases in obesity, type 2 diabetes, metabolic disorder in those individuals that were gestating during that starvation period.
Yeah, there's this idea that you mentioned in your paper called the thrifty phenotype, right? Which is that mother is going through a very hard time energetically. There's not enough to eat. And it sets off this series of developmental events where it prepares the child for an environment that has a lot of energy scarcity. So there's not going to be a lot of food. So in the case of the Dutch, they have this Dutch hunger winter. World War II ends. Food becomes very plentiful, but the kids' metabolic systems are geared towards a food scarce environment. And when those two things are crossed, you end up with more metabolic diseases.
Yeah, exactly. And the mechanism for this is presumably epigenetic effects.
Right.
And so Yeah, a lot of my work subsequent.
We were like, this is epigenetics, and then I didn't describe epigenetics at all. Um,
Yeah.
Epigenetics is like this idea that the way that your body expresses like genes and proteins, like all that stuff, it's not just like an on or off switch for the most part. It's more of a dimmer switch. So like
That's great. Yeah.
yeah, so like you can like turn the dimmer all the way on or you can turn it all the way off, but most of the time it's like kind of somewhere in the middle, and it can like go up and down depending on your environment, but it can also like the preset level that you have, a lot of that it depends on like your mom and like the environment that she was in.
Yeah.
So that's like how this mechanism for how it's supposed to, how epigenetics happens. It was so eloquent and then it wasn't.
No, that was great. That's exactly how I like to describe it too, is the dimmer switch. And I usually say that, like, allelic variation, which is usually what we think about as genetic variation. So differences in the letter of your genome in a gene that changes the way the protein functions, right? Like that's one way that you can kind of set a difference in what kind of trait you're going to have specifically. But then, yes, the epigenetic element is where you toggle that level as well. So like those are like two really interesting sources of variation in a trait.
Yeah. Yeah. It's really cool. It's like sometimes people are like, why study monkeys? Like, what's the point? And it's like, they're actually really great models.
Well, yeah, they're great models. And in addition to that, like, these monkeys in particular, it's not like it would be completely horrific to enact a study like this in humans. Like you just couldn't do it. And so you have events like the Dutch hunger winter, and also like there was a flood in Bangladesh that also was being people there were being studied at the time when the flood happened, and that led to a famine. And that also is a data set you can use. It's completely opportunistic, but you lose an element of control that you could have with captive animals like non-human primates, where you've got the same or very, very similar genetic background. And part of why I did this work too is work had already been done in that colony showing that when you get obesity in these monkeys, it looks like metabolic disorder in humans insofar as the biological systems go. So yeah, you can learn quite a lot about events that in humans would be horrible tragedies, but in a captive primate, it's like a model system to find out how you can actually understand the impacts of these events on human bodies.
Yeah. Yeah, especially with these particular monkeys, like it's not like a horrific tragedy befell them. They were just switched. Someone just switched their diets because they were getting too chunky.
Yeah. They just decided they weren't gonna eat a lot of food. Yeah, exactly.
Which is a bummer, but it's different.
It's a bummer, but it is very different. Exactly.
So I actually also wanted to ask you, because you have started more recently working on another monkey, which is the yellow-tailed woolly monkey.
Oh yeah.
Which is endemic to South America, like Peru mostly.
Peru, Andean Peru specifically. Yeah.
Yeah. They're pretty cool looking monkeys.
They are. Like I joke with my students, they're this like really beautiful mahogany color, and actually it didn't really work in their favor, because for a long time part of why they become critically endangered is people were using them as like saddle blankets, because their fur is very lustrous and beautiful. But they also, like the vervets, have very interesting genitalia, and I joke with my students that like that's why I study these two species. They have to have colorful scrota, because the yellow-tailed woolly monkey, in addition to like having a yellow patch on the tip of their tail and like a little bit of yellow in their muzzle, they also have like this, it's called a mechon in Spanish, which I think is like a very satisfying word to say. It's a bright yellow scrotal tuft. And like when I say scrotal tuft, it's like a huge ball of like golden fur surrounding their scrotum. And the females also have around their clitoris, it's more like a wispy little mustache of golden fur. So really, really like kind of funny, also kind of beautiful, like really either way, very striking monkeys.
That's so interesting. Monkeys were not it for me when I was in grad school. I'm pretty scared of them. Monkeys not as much, but like definitely chimpanzees. Like I am.
Oh yeah, no, I'm terrified of the apes. Like I would much rather work with monkeys that are of a manageable size.
Monkeys of a Manageable Size is my band name, actually. No, No, but so I like I haven't really like spent a lot of time like thinking about them or interacting with them. And like as I've talked to more primatologists on the show, I'm like, actually monkeys, like non-human primates, they're actually pretty cool. I was wrong.
Oh, they're super cool. Yeah. No, I could watch them all day. I mean, I have watched them all day, many days of my life.
So you're also studying these monkeys and you're like
Yeah.
studying range. One of the other themes that I see in your work is a lot of genetic studies,
Mm.
which is really cool. This is something that wouldn't have been possible like 30 years ago, 20 years ago.
Oh yeah, no.
Yeah. Like I
No.
still remember when they sequenced the entire human genome. I was in high school.
Yeah, me too.
Yeah. And the teacher came in and was like, you will not believe what happened.
Yeah. And it's been interesting because in the course of my career, genetics has just become so much more, I don't know, complex and interesting. And you've been able to do so much more. Like when I was in grad school, we were still using microsatellites. We were doing like what we call Sanger sequencing, which is, very good sequencing, but of like very small fragments of the genome. So like maybe 200 or 400 base pairs at a time, 200, 400 letters. And microsatellites are basically like these little repetitive areas of the genome that you can use for, you may have heard it called DNA fingerprinting. And yeah, I was a grad student of Tony DiFiore, who's at NYU. He was at NYU at the time. He's now at UT Austin. And, we're in the genetics lab, it was like, we would extract the DNA from the poop that we collected and like, we could do some microsatellite stuff. We could maybe sequence a little fragment of the mitochondrial genome in order to kind of, do population genetic studies. And then, after that, when next generation sequencing started happening, all of a sudden the game just completely changed. So by the time I was in my postdoc, people were like, well, why would you want to do microsats? Like, why not just sequence the whole genome? It's so crazy that within a 5-year period, we went from, microsats to that. It's really incredible.
I think the other thing to emphasize, I've been thinking a lot about this kind of stuff lately because I cannot emphasize enough how much like microsats and Sanger sequences were amazing.
Yeah, yeah.
When we were there, right? It's like if maybe this is also too much of an old person reference, but like if you look at your photos from like before iPhones and not like the click ones, like I had a shitty old Samsung phone.
Mm-hmm.
And I was like, wow, this is like breathtakingly detailed. And now I look at it, I'm like, this was taken on a potato. Yeah. Or it's like video games, like the video games that you played as a kid, you're like so detailed. I'm so immersed in this world, like Zelda.
Mm.
And then you look at like Breath of the Wild and you're like, this is a completely different game. This is crazy. But at the time, it was so amazing. Anyway, I've been thinking about this a lot lately. I think it's because I work with a lot of 23-year-olds and I'm like, your experience of the whole world is just so different. And I'm sure that this is all going to happen to you in like 10 years. Like you're going to look back at like these stupid still photos that we have on our cameras that aren't like 3D immersive or whatever. And you're like, it was the dark ages.
Oh yeah. No, it was funny. I was teaching about the Scopes trial in class yesterday, and I was like struggling to articulate to the students how like radical it was that they had like live radio broadcasts at the trial. And I was like, I don't even know how to make this sound as amazing as it probably and like mind blowing as it probably was at the time.
Yeah. Yeah. It's okay. They'll age and they'll understand one day.
I know. Yeah, exactly. Give it time. Give it time.
But no, it's really cool. And it's cool that you have like all of this genetic data. And like one of the things that I see in a lot of your work is a lot of statistical work. So this is my once a show plug for doing math.
Yes, please do. Please learn math. Please learn how to code in R.
Yes.
It's amazing. Like until I taught myself R in grad school. And prior to that, statistics was just like a class that I took and I had no idea how to do anything. And now like to be able to just like get data and analyze it is just so mind blowing to me. I love it.
It's so satisfying. This is probably another experience that's going to make me sound really old, but it's so satisfying to be able to be like, I have a question and I can write a code and immediately answer it. And that's so cool. It's like the equivalent of like when we got smartphones in our pockets and like you'd be like, I have a question about, can my dog eat cantaloupe? I can just Google it right away. I know the answer. I don't have to go to the library. I'm sounding so old right now.
No,
Uh,
I'm right there with you. I miss, as part of me misses it, like, because everything was a lot slower, which was, it was both good and bad, right? Like, you could be digging into a question for months, and then somebody publishes something, and you're like, oh, okay, like I guess somebody was already thinking about that, or it can take you a long time to find the answer to a question, or whether or not a question has been asked and answered, and things just move a lot faster now, which is great. But there was a bit of satisfaction to like
that's true.
getting to the end of that just by digging through a card catalog.
Oh my God, I do miss that actually.
Yeah.
Okay.
Okay, so
Nostalgia
we're old.
moment
We've
over.
established that.
Do math. That's really important.
Do math. Yes, that is the point. Do math, learn R.
Learn R. Um, I was going to ask you what it's like for you to be in the field actually, because you have spent
Oh, yeah.
time like out. Well, what is that like? Is that enjoyable? Like what are things that you think that would help people like the field? Like
Yeah.
if you're someone, for example, that really needs a flush toilet, is the field for you?
It can be. It can be. So yeah, I think I've spent 8 years of my life doing field work at this point, if you add it all together. So I've done a lot of field work in a lot of different conditions. And I think whether or not you enjoy field work or whether or not field work is for you, I mean, it depends on a lot of things. So I mean, I'm gay, I'm queer. And so field work for me, like I was definitely going to do it when I was much, much younger. I was like, this is what I want. I'm going to do it. I had camped a lot. Like I was pretty outdoorsy. I was also really into the science of it. So I was like, this is for me. And, depending on where it was, sometimes I had to be closeted and that was really unpleasant. On top of that, like, when you're in your early 20s and you're like at a field school or a field assistant, all the straight people are hooking up with each other and you're just kind of like, I will be the chaste gay man in the forest, which is also not super, not a great way to spend your early 20s. So yeah, there was also a lot of like nervousness, like what would happen if I was outed or if somebody figured it out. Men would ask me what I think about women, and I'd be like, oh, I'm too busy with my work to think about that. Um, so yeah, there were like some struggles related to that, and of course, like I do a lot of activism around this too, just making people aware of the challenges that queer people face in the field, and of course for trans folks, there are unique issues. For women, there are unique issues. For people of color, depending on where you're going, there can be also unique issues. So yeah, like I think almost anybody can find a field experience that will work for them. It really depends on whether or not they can find that experience. Depends on whether the principal investigators at that field site make it legible to them as a place where they can actually have a good field experience. And this is something that I advocate a lot for too. Like I've been talking about this with National Geographic. They have a queer and allies hub, and me and a large group of other Nat Geo funded field workers have been trying to help them to think through guidelines and ways to kind of think through how to help people feel that they can go and do field work regardless of their background or their visible identities or hidden identities. And I would say, yeah, so I think a big element of that, and this comes from, I mean, you mentioned that you just interviewed Kate Clancy. So one of the studies that she kind of spearheaded with Robin Bernstein and Katie Hind and Julian Rutherford, the SAFE study, what they really found in a follow-up to the study, and for context, the study found that like sexual harassment was rampant at field sites.
Yes.
Most female identified people that were in that study, I think it was something like a ridiculous number, like 70% had like either witnessed or experienced harassment. And not insignificant proportion of those individuals had experienced it themselves personally. In their follow-up to try to find out what it was about field sites that led to harassment or not, what they found was that field sites that had really, really clear codes of conduct with very clear articulations about what would happen if somebody engaged in harassment, those were the sites where harassment wasn't happening. And so I do think that, that one element at least of field work is something that we as researchers and the people who are like running these field sites, we can make sure that those sites are welcoming to more people than they have been historically. And so that the people that go there don't have to grit their teeth through it. They can actually go there and not have the kind of cognitive load of worrying about harassment or worrying about being outed or worrying about how people are going to take them, they will know that there is like a recourse if somebody does do something that makes their experience less than good, right? So I mean, I think that's kind of a meta a meta answer to what you're asking.
No, I think it's worth talking about it. One of the things that we also talk about periodically on the show is like how to pick a grad program where you won't regret your entire life.
Yeah, yeah.
And this is kind of along those same lines, right? Like how do you
Yeah.
find a field school where you're going to be psychologically safe and so you are able to do your work?
Yeah. No, I think that's part of it. Like, I mean, obviously, the Whisper Network is always a place to go to find out for sure. And this is something, so the American Association of Biological Anthropologists, we have a queer group there, our LGBTQIAA plus interest group in the Committee on Diversity. And the members offer ourselves up to like talk to anybody about this, about various field sites, various countries, what it's like to work there, whether or not it's safe for different identities. And usually if you reach out to one of us, we'll connect you to somebody who can answer your question about that. Because yeah, like in the field sites that I've worked, increasingly it's, and this is part of this is just the world changing, and part of this is the countries where I'm working. I've not had to closet myself in the field, which has been really great for my mental health and for just, being able to get my work done. But there have been places where I have had to be very deeply closeted. So I did some work in the Gambia, and this is when Yahya Jammeh was president, and he was on the radio telling people to kill gay people and that, gay people didn't belong in their country. And I was working there during that time, and it was terrible. I
Yeah.
mean, I couldn't be out. I was also going through a breakup, like a long distance breakup with my boyfriend at the
I'm so
time, kind
sorry.
of. And so yeah, it was just like really a horrible experience. And so like I won't work there again just because I have bad associations with it. And the government is different, I'm sure, like, and there might be other queer people that get along just fine there. But for me, it just wasn't for me. And there are other sites where it's been fantastic. So like, for example, part of the reason why I started working in Peru. I mean, yes, the yellow-tailed woolly monkeys and their golden scrota are really wonderful. But in addition to that, I went down there in 2019 to give a talk with the Peruvian Primatology Association. And my husband came with me, my now husband, and we went to help teach a field course in Cochacashu. And when we got on the boat, we were introduced as boyfriends. And and we got to the field station, we were introduced to all the students, predominantly Peruvian, that we were boyfriends. And like nobody cared. And it was so just like so different from when I started out in the field, like feeling like I needed to be closeted, really worrying about it. And to be clear, like maybe I didn't have to be worried. Like maybe that was just kind of me being having an overabundance of caution. But as a queer
Maybe
person, like better that than going in blind and just being out and then facing challenges for it.
not to harp on the like we're old thing, but I think unless you lived through the early 2000s, like the casual homophobia that was like
Yeah.
everywhere it's hard to explain.
Yeah, no,
It's
exactly.
like it's like for example, if you watch old episodes of Friends, sometimes you're like
Oh, no, it's terrible. The
you're
show
like,
is awful.
excuse me, it's a really bad
I
show.
know when I hear my students watch Friends, I'm just like, really?
I know it's like please for the love of God, not
Yeah,
that I'm trying to be
something that.
snooty. Anyway, but like I don't think that you were in the. I think that you did what you needed to do to feel safe, and that makes
Yeah,
sense to me.
exactly, exactly, and that's the thing, right? Like, if there are cues that make somebody queer or somebody black or somebody trans or a woman not feel safe at a field site, and the PI is just completely unaware of that, then that field site is going to continue to have problems.
And sometimes the PIs are the problem.
And sometimes the PIs are the problem. Yeah, exactly. And so, yeah, it really, like, I do feel that there's obviously been a huge culture change in the ABA surrounding all of these issues, which is fantastic. This doesn't mean that every field site of a biological anthropologist is fantastic. There are also people who are really well-meaning who just don't know a student of a particular identity, and they don't share that identity, and it's just completely off their radar. But the resources I think are now out there and in the public in the publication record, thanks to Kate, Robin, Julianne, and Katie, and everybody else who's worked on these issues, especially in the last couple of years around queer issues in particular, like I do think there's not really an excuse anymore to not think this through
Yeah.
and not to try to make your field site more friendly to anybody who might go there. But having said that, I also have to acknowledge, like I mentioned, the Gambia and that field site, there was nothing that we could do to make that field site comfortable for me. Like nothing would have helped.
Right.
And there are, field sites where, I have colleagues who work in certain countries or certain contexts where they can't be out, and that's where their study species is, and that's what you have to do in order to do the science you want to do. So largely, it's also like a personal decision for those of us who have identities that might be uncomfortable or in danger at certain field sites, and it's up to us to like really think about whether or not that's something that we want for ourselves. And when I was in my 20s, I was like, absolutely, like I'm going to go do the science, like I'm going to be that queer person, and if it means I have to be closeted, so be it. And nowadays I'm just like, yeah, no thanks. Like, I don't
I'm
really
going
want
to
to be that
Peru.
closeted. I'll go to Peru where like people are okay with gay, right?
And the food's really good in Peru.
And the
So
food is amazing. Yes,
yeah.
Lima, incredible, incredible food.
Which is really solely how you should pick a field site.
Oh yeah, I mean it's a non-negligible factor.
um, I was wondering, one thing that I do ask everyone who goes out in the field, what's like one thing that you take with you out into the field that's like not on anyone else's packing list? You're just like, this is something that's really important to me that I take every time when I go out. Like, for example, someone once told me that they bring their pillow from
Hmm.
home. They like, I have to have my pillow. Or someone told me that they bring a very specific type of coffee.
Uh-huh.
Another person is like, I bring 4 pairs of glasses because you break one pair of glasses in the field and you you never come unprepared again.
I bring a Kindle loaded with gay romance novels, like trashy gay romance novels. And this is something that, yeah, that I started to do when I still had to be, moderately closeted in the field. And I just kept it going because, when you're in the field, you're not necessarily, I mean, I guess as a PI, I could make my field site gay culture. But you're still not necessarily like, steeped in it, and it's not as accessible as it might be otherwise. So I like to bring the gay romance novels just to like be gay in a way that if I need it to be unobtrusive, I can be unobtrusive about it, but I still am plugged into that element of my life.
Totally. I also think that when I travel and I go places that aren't like that don't have the best internet or like whatever, I read so
Mm.
much more.
Oh yeah, yeah, exactly.
I too have gotten on the Kindle train. My mom always she doesn't get upset, but she's like, I don't understand how you read so much. It's like, because I go on vacation and I go camping and I read 13 books in a week because there's
Yep.
nothing else to
Yeah.
do.
Yeah, exactly. And when you're on a boat or when you're on a plane, like in the field, I mean, I was going to say like my field experiences have changed so much from, when I was, doing my dissertation and I'd be out before sunrise and until after sunset with the monkeys, collecting data, collecting poop. Now that I'm like a PI, I'm like, I spend almost all of my time just like making deposits and traveling between field sites to make sure that things are going well. And I think I was in Peru for a month this past summer, and I spent literally just one day with the monkeys, and all the rest of the time I was like talking to the community, talking to assistants, talking to collaborators, and just making sure that things are going smoothly. So yeah, the experiences change a lot, but it also means I travel a lot more when I'm in the field. So yes, having something good to read is absolutely essential.
That's a really good answer, I think. And it actually leads in perfectly to my last 3 questions, which is
Mm,
if someone wanted to learn more about either your work in particular or kind of like the basics of the kind of work that you do,
mm.
where would be a good place for them to start?
I mean, I really think that, oh my God, I had an answer for this like before I came on and now I've completely blanked on it. Um, I think if you're thinking about like life history theory and epigenetics, I think to be honest, I really love the primary literature. Like I wanted to recommend something that's like really accessible.
There's for epigenetics, there's a book by Nessa Carey. It's called The Epigenetics Revolution,
Let me write that down.
which I read. It's older now because I read it right after I finished grad school in 2015.
Yeah.
But I thought it was a really good explanation of epigenetics. I thought it was like a really accessible introduction to it. If that helps, I just answered your question for you.
No, that's great. I'm clearly blanking.
No, it's okay. It's okay. I know. Sometimes it's like we put people on the spot, and like, there's been a couple times where I've asked people, and they're like, "Excuse me," and they turn around, and they just like rifle through their bookshelf.
I know. You must have noticed. I'm like staring at my bookshelf right now to try to think through like what I might recommend. I mean, yeah, I'll have to rethink on this, but I think your suggestion sounds great.
Oh, thank you, thank you so much. I'm like looking at my bookshelf now too, and I'm like trying to think of like, do I have one? I think my life history books might be in my bedroom. All I have in front of me are cannibalism books right now.
Well, we can always go back to the original. So like Stearns
Hmm.
with 1992, 93. Like I thought that was like a really accessible description of life history theory. And it's like one of the early ones. So like a lot of things have been elaborated on that and built up of that. So I guess I would recommend Stearns.
Yeah, yeah. No, I think that's a great recommendation. And honestly, like, it's not like it's hard to find. Like, if you get on Google Scholar and Google Life History Theory and then sort by number of citations, that's a great way to get started.
Yeah, exactly. And like just for context, what I'm recommending is The Evolution of Life Histories by Stephen Stearns, which is apparently on Google Books and it's also available on Amazon. So it's around.
Perfect. What are you reading for work right now?
Yeah. So for work right now, I have a PhD student, Jimmy, who's thinking about doing something that Trudy Turner and I have been talking about doing forever, which is actually leveraging our genomic data to do a taxonomic revision of all of the savanna monkeys. So, like green monkeys, vervet monkeys. They're this really weird taxon where below the genus level, we don't really know what's going on. They all can interbreed, but they also have population specific things going on. And that makes taxonomy really confusing. And the history of their taxonomy is very confusing. So I've been reading some like really old school vervet monkey taxonomy papers like Dandelot 1907 and trying to like refamiliarize myself with the older literature so that Jimmy and I can kind of work through what we can say with the genomic data.
I will say taxonomy is wild, just like as a thing.
Oh yeah.
Because like, it's one of those things that in high school they're like, it's so simple. This is what a
Oh
species
no.
is. And then like you're like, it's like as soon as you get to college, they're like, we know nothing.
Yeah, exactly. Like the boundaries are so blurry, like they're completely arbitrary. It depends on what you're trying to do. Yeah. So we're gonna make an effort on it over the next year or so. So that's what I've been reading this week.
That's really cool. That's really interesting. It's always fun to go back to those really old papers because it's the writing is really different. And I think it kind of gives you like a window into a very different world of anthropology.
Yeah, very much so. And what I really love about it is I've read some like really interesting ideas in some of this old literature that like basically like presage some like cutting edge stuff that people have done in the last 5 or 10 years. And I'm like, oh my God, they were already thinking about that back then. And they actually had a really well articulated idea for how this worked. I think it's really fantastic to go back to that writing because it's really kind of generative.
Yeah. Yes. Yes. And then last question for you is, what are you consuming for fun right now? It can be books, it can be whatever, honestly. Some people say kayaking. Whatever floats your boat.
What floats my boat right now? Well, when it is snowing, I live right next to Franklin Park in Boston, and so if it snows, I can go cross country skiing there in the park, like right next to my house, which is amazing, and I
That
love
is
it.
such a Wisconsin answer.
Ironically, I never went cross country skiing when I was living in Wisconsin. I only started doing it after I'd already left.
That sounds great though, honestly.
Yeah, I love it. I love it, and then it's between that or if it's like too cold outside, then I watch really trashy gay movies, like B-level movies. I don't know if you've heard of Breaking Glass Productions or
I
like
haven't, but I'm
there's
gonna
this.
look it up now.
It's like a certain kind of of movie that Breaking Glass distributes that I just like. It's like gay romance novels. Like I just can't help myself. I just devour them.
Yeah, I've talked pretty extensively about how much I love trash on the show. Like
Yeah,
you don't have to think hard. You just it's like eating a bag of chips. It's just good.
exactly. And this is maybe why I study vervet monkeys because I'm sure they would agree with that.
Intellectual trash, literal trash, you know.
Literal trash, same difference.
Well, thank you so much for your time. I really appreciate it and have a wonderful day.
Yeah, thank you. You too.
Thanks for joining me for this episode of the Anthrobiology Podcast. If you have questions or comments, you can reach me at gabby.lapera@anthrobiology.com. That's spelled GABY.LAPERA@anthrobiology.com. This show is produced by me. Music courtesy of Chad Crouch, aka Poddington Bear at Sound of Picture.